We've truly moved away from the gram-negative bacilli, and we are talking about a completely different group of organisms in this lecture. So today we are going to talk about the Mycobacteria organisms, and this lecture has been separated into 5 parts. So there are many different organisms, species of Mycobacterium. There are approximately 100 documented Mycobacterium species. Of course, we are going to focus on those that cause human disease. So we're not going to talk about every single solitary species. In the next few slides, the organisms that can cause disease are mentioned. So we have our pathogens over on the left of this slide, which include Mycobacterium tuberculosis, Mycobacterium bovis, Mycobacterium ulcerans, and Mycobacterium leprae. We have another group of Mycobacterium organisms that are often pathogens, not maybe as common or notorious as those on the left-hand side, but these are Mycobacterium genevents, Haemophilum, Consacii, Marinum. There is Mycobacterium avium complex and Mycobacterium xenopi. On this slide, on the left, we show the Mycobacterium organisms that are potential pathogens. Don't usually cause disease, but can be opportunistic, cause opportunistic infections. These are Mycobacterium fortuitum, colonii, abscessus, malmonense, simii, scrofulation, and Schulgi. Now on the right, there are very rare pathogens. These organisms have been documented in some diseases, but it's not common at all. So these organisms are more commonly environmental organisms. So there's Mycobacterium gordoni, flavescens, gastri, non-chromogenicum, terrai, flea, smegmatis, vaxi, and thermoresistible. Now those that are colored in blue, those organisms are part of another complex in Mycobacterium, which is the Mycobacterium terrai complex, which are environmental saprophytic organisms. So the general characteristics of the Mycobacteria. There are 100 recognized species. These organisms are bacilli. They're categorized as gram-positive bacilli. However, they are highly resistant to gram staining. The cell wall has a very high lipid content. It's loaded with mycolic acids, so that's why gram staining is not commonly used for Mycobacterium. In order to stain Mycobacterium, we use what's called acid-fast staining. Now, we talked about acid-fast staining early in the semester when we were talking about other gram-positive bacilli with the nocardia, the branching organisms. So some of those organisms can be stained with a modified acid-fast stain. So Mycobacteria, you use the non-modified acid-fast stain, and they require complex media for growth. So Mycobacteria are usually not grown with your standard laboratory media that you would use in to grow up, let's say, E. coli. These organisms are strictly aerobic. Many of them that are associated with disease are very slow growers. They take 2 to 6 weeks to produce colonies on a plate. Mycobacterium tuberculosis, one of the major disease-causing species, has enhanced growth if you incubate it in 5 to 10% carbon dioxide. And another pathogen, Mycobacterium leprae, fails to grow in vitro. So you wouldn't grow that organism on agar or broth artificial media. Now, we already said the Mycobacterium cell wall has these layers of lipoarabinomannan, mycolic acids. It is a gram-positive organism. It does have the peptidoglycan, but it does have a very different cell wall than your other gram-positive and definitely different than your gram-negative bacteria. So that lipoarabinomannan functions very similarly to lipopolysaccharide in gram-negative bacteria. Again, we have that thick mycolic acid layer that the gram staining doesn't really get through very well. So we use acid-fast staining to stain Mycobacterium, and there are two different types of acid-fast staining. One is the Ziehl-Neelsen, and the other is the Kinyon's modification. So the Ziehl-Neelsen acid-fast staining method requires a heat source to force the stain to go into the organism. The primary and secondary stains are carbofuscin and methylene blue, and the organisms stain a red color. Even though they are not gram-negative rods, don't confuse the red color of Mycobacterium with a gram stain. They're very different. So because Mycobacterium is red, has nothing to do with it being gram-negative. It is a gram-positive bacillus, technically. Now, the Kinyon's modification would be normally used in a micro laboratory because you don't need the heat source. So whenever you could stain something without using a Bunsen burner or any type of flame in a laboratory, that's the way you're going to go. So the Kinyon's modification method uses carbofuscin, just like the Ziehl-Neelsen, but it uses malachite green as the secondary stain. Now, Mycobacterium is also stained with a fluorescent stain called auramine rhodamine, and a lot of different bacteria can be fluorescently stained with auramine rhodamine. And for clinical laboratories that have fluorescent scopes, this would be the staining method of choice. So here is an acid-fast stain of Mycobacterium tuberculosis, and with a Ziehl-Neelsen stain, the background would be a blue color and the organism stain this bright reddish pink color. Same with the Kinyons, but the Kinyons, the background is a green color and the organism stains this magenta fuchsia color. So for safety, Mycobacterium tuberculosis is considered a biosafety level 3 organism. Some of the other Mycobacterium organisms, such as the Terrae group, are not biosafety level 3 organisms. So most of the Mycobacterium, you could just use standard precautions, your typical biosafety level 2 precautions, where you would wear gloves, you would use a disposable gown, washing your hands. But Mycobacterium tuberculosis requires a biosafety level 3 lab, and it requires you to wear a respirator. So there are N95 and N99 cloth respirators that you can use to work with the organism. Now, people that work with Mycobacterium tuberculosis, or anyone working in a hospital, so for those of you that are going to be starting your clinical rotations, and anyone who is currently working in a hospital or will be, you have to get a TB skin test annually. I used to do research on Mycobacterium tuberculosis, and I had to get skin tested every 6 months, and we'll talk about the skin test a little later in this lecture. So to work with Mycobacterium tuberculosis, you would want the lab to be separate from other labs. So you will see, for those of you that when you're going through your micro rotations, if you haven't been exposed to a clinical micro laboratory yet, In the clinical laboratory, you'll have different sections. So there'll be a main section where you'll do a lot of processing. There'll be a section with your instrumentations like your microscan or your Vitek, but usually there's a separate lab that has a door that closes that will be the tuberculosis laboratory. So Mycobacterium tuberculosis is always worked on in an area away from all of the other areas of the laboratory. There should be a non-recirculating ventilation system. The lab should have negative air pressure. All work should be done in a biological safety cabinet that is certified annually. And of course, as with any other organisms, you have to use appropriate disinfectants. Now, there are special sprays for Mycobacterium tuberculosis because it has that thick mycolic acid cell wall. It does need harsher disinfectants, so they do sell special sprays that if you are working in the TB area of the lab, likely that those sprays would be used to disinfect the bench tops and the inside of the cabinet. So specimens that you will get as a clinical microbiologist for processing, well, for tuberculosis especially, and for your MAC, and for all your organisms that cause disease, the most common specimen is going to be an expectorated sputum. So someone coughs up from deep into their lungs into a container, and that's going to be the specimen. When I worked in the clinical microbiology lab, that's the specimen I liked working with the least. I really disliked sputums. They're kind of gross. I will work on a stool any day over a sputum. But you will see a lot of them, especially for TB testing. Other respiratory specimens include normal saline, the normal saline nebulized induced sputum, where they'll actually insert saline through a nebulizer to generate some sputum. There are transtracheal aspirates in individuals that cannot cough themselves, cough up sputum. They'll go through the trachea and aspirate a respiratory specimen. There's bronchoalveolar lavage, which is a much more invasive type of specimen because the lungs are actually washed and pulled out. You pull out samples, so that's not a specimen that's going to be done as often. You're going to get the sputums more regularly. And then there could be a laryngeal swab. Not common for TB, but sometimes you might get one. Now, TB can spread throughout the body, so you could get a body fluid that you would need to test for Mycobacterium, a pleural fluid, a pericardial fluid, a joint aspirate, gastric aspirate, peritoneal fluid, cerebrospinal fluid, a stool, urines, and pus, depending on what type of Mycobacterium infection you're dealing with. Also, you could get a body tissue specimen, blood, bone marrow biopsies, aspirates, solid organs, lymph nodes, bone, and skin. Now, some of these specimens might need some processing. So uncontaminated specimens like blood and cerebrospinal fluid wouldn't really need to be processed as much, but they may need to be centrifuged. Usually you want to centrifuge a sample that should be sterile, because if there's only a few organisms in there and it's diluted out in the fluid, you might miss it if you then go to stain that specimen. So you want to centrifuge it so that you pull any bacteria that might be present down to the bottom, you pull off the supernatant, and you stain and do a culture on the sediment. So for uncontaminated specimens, your processing would be centrifugation. For contaminated samples, such as your respiratory samples like a sputum and a stool sample, you need to both digest and decontaminate that sample. So you use things like N-acetyl-L-cysteine to digest that mucus, so that the sputum is usually thick mucus. So the N-acetyl-cysteine is known as a liquefying agent. So it's going to digest that mucus and make it less viscous. Then you have sodium hydroxide, which will digest and it will decontaminate. So it will get rid of some of the normal oral flora that's going to be present as the sputum comes up through the oral cavity, but that processing will not damage Mycobacterium if it's present due to that thick, waxy layer. So digestion and decontamination, and you could read about this procedure in more detail on pages 590 to 591 in your textbook. But you can use the sodium hydroxide at 2%, 3%, or 4%, which again is a decontaminating agent and a digestant. N-acetyl-L-cysteine is that liquefying agent that digests the mucus. There's also benzalkonium chloride, which is trade name is Zephyrin, with trisodium phosphate. So this is known as ZTSP, which liquefies sputum very quickly, but you have to expose to the decontaminating agent for long periods of time. Also, Zephyrin is bacteriostatic to Mycobacterium tuberculosis, so you don't want to inhibit the growth too much, because if you're culturing your specimen, you don't want to stop the growth of that organism. Oxalic acid at a 5% concentration can be used to decontaminate specimens that might be contaminated with the organism Pseudomonas aeruginosa, which can be an upper normal flora of the upper respiratory tract. And remember, Pseudomonas aeruginosa is a common cause of infection in people with lung diseases like cystic fibrosis. In order to differentiate your Mycobacterium, the things you need to consider, and definitely keep this in mind as we go through the Mycobacterium lecture, because you as clinical microbiologists are responsible for differentiating one organism from another organism. So the rate of growth. The various Mycobacterium organisms vary in their growth rates. The colony morphology, as usual, we like to look at the colony morphology, colors, patterns. Pigmentation. Some of the Mycobacterium tuberculosis organisms produce pigments, others don't. The nutritional requirements, the incubation temperatures, the optimum incubation temperatures, as well as biochemical testing. The types of media that you are going to use to culture Mycobacterium tuberculosis. One of the common medias is an agar slant that is Lowenstein-Jensen. It's a very pretty agar. It's kind of a mint green color. There's also Petrignani. There is the ATS, or American Thoracic Society. There's Middlebrook, 7H9, 7H10, and 7H11. These are broths and agar media. There's Gruft's modification Lowenstein-Jensen media, and there's Mycobacticell Lowenstein-Jensen media. So depending on what laboratory you're working in, they might use one of these media, or they might use a combination. So if you're doing colony counting, you want to use a lighter colored media like Middlebrook 7H10 or 7H11. If you're just trying to grow the organism, see if it's there, Lowenstein-Jensen is a good media because it's nice and rich, and Mycobacterium really likes to grow on it. So your broth and solid media, there are the egg-based media, like I already said, Lowenstein-Jensen, very rich media. It contains malachite green to inhibit oral flora contaminants, and that media, that's why it's that mint green color. It has malachite green dye in it, and it has egg in it to neutralize any toxic materials. Non-selective media such as Middlebrook 7H10 or 7H11, and this is a more clear media, and it's non-selective. It doesn't have any antibiotics in it or any dyes to inhibit any contaminants in it. And then there are selective agars that have specific antibiotics added to them to decrease that normal flora contamination in your sputum samples, especially to increase your recovery of your Mycobacterium. So here are some different Mycobacterium organisms. On the left-hand side, you could see an organism that is pigmented. It produces a nice bright yellow color when it grows an agar. On the right-hand side, this would be more typical of a Mycobacterium tuberculosis organism. It is non-pigmented and what's called buff colored, and it kind of has a very brainy look on agar media. It kind of heaps up on top of one another, a little bit wrinkled. Mycobacterium tends to be dry, very dry colony. Now, there are plenty of other media that can be used, and there are a lot of different systems specifically to identify Mycobacterium tuberculosis. And every clinical laboratory uses a specific tuberculosis system, and they're going to vary from one lab to another. So I don't like to focus on a specific system because it just depends on where you're working, what system they're going to use. So some of these systems are the SeptiCheck AFB, which stands for acid fast bacillus system. There's the Midget or the Mycobacteria growth indicator tube broth system. There's the BacTech TB system, the MB BacT system. So the SeptiCheck, the Midget, and the BacTech are very commonly used among clinical Mycobacterium laboratories. So here's the SeptiCheck acid fast bacillus system. So this is a biphasic media system to both detect and isolate mycobacteria. So traditionally with Mycobacterium, you'd have to inoculate on your media. You'd have to wait possibly 4 to 6 weeks for it to grow, and then you'd have to do all of your tests on it. These systems are much more rapid because you're not relying on growing the organism first and then doing tests. Here is the BacTech system, little bottles. You sterilely inoculate your sample into these bottles and then put it into the equipment. So these systems are likely what you will use in the Mycobacterium laboratory, but you still may culture some Mycobacterium, so it's nice to know about the colony morphology of the various species. There's also staining. You may not stain that often with acid-fast stains because you're going to use the systems more commonly to diagnose, but you do need to know how to do an acid-fast stain. Very similar to gram staining, very similar technique, just using different dyes and different decolorizers. So here you have your an abscess aspirate that is actually gram stained, and you can see gram positive beaded bacilli. So that is the picture on the bottom. The picture on the left is also an abscess, and this is a Ziehl-Neelsen stain. So you have your Ziehl-Neelsen staining and you have your gram staining. Now here you have your auramine rhodamine staining or your fluorescent staining, and on the left hand side you could see the rods very bright, the fluorescing under the fluorescence microscope. And on the bottom right, this is a skin biopsy stained with Ziehl-Neelsen stain from a leprosy patient, and you could see the organism staining that that bright pink color. Pigmentation is also incredibly important with your Mycobacterium species. So some of these organisms produce pigments and some do not. And then the ones that do produce pigments produce them under specific conditions. So we have our photochromogens. These are the Mycobacterium species that produce a pigment only when they are exposed to light. So this slide is really important. You have to know the difference between the photochromogens, scotochromogens, and non-chromogens. So photo, light. That's easy. Organisms that produce pigment when they're exposed to light. Scotochromogens produce pigment whether they're in the light or the dark. So these are organisms that are always going to be producing a pigment. It doesn't matter if they're dark, sitting. How we used to test for pigmentation way back in the day was you would take Lowenstein-Jensen slant, you would take your specimen and inoculate 2 slants. One of those slants you would keep exposed to the light in the incubator, and the other slant you would wrap up in tin foil so that absolutely no light can get through it. And, a week or 2 later, you would pull off your tin foil and you'd see if neither one of them produced pigment, it would be a non-chromogen or non-pigmented organism. If the one that didn't have the tin foil on it was pigmented, but the one that was wrapped up in foil didn't have a pigment, it would be a photochromogen. And the one that, if you had a pigment in the one exposed to light and then you pulled off your foil and there was also a pigment, that would be a scotochromogen. So here showing some of the Lowenstein-Jensen slants with the tin foil wrapped around to block the light, and this was the classic way to test for pigmentation. So we're going to move on to the second part of the Mycobacteria lecture.